Welcome to my channel Adhyan with Flow. Chapter 14, Respiration in Plants. Yes guys, plants can also respire. Look at here. We know at a daytime what they do. They do photosynthesis. We know all this thing that at a daytime plant perform photosynthesis. What is the reaction? CO2 plus H2O. They make food and release some oxygen in atmosphere. Now, at a night time, yes, plant perform respiration. Yes. And what is the reaction of respiration? Now, this oxygen take from the surrounding, breakdown of the food, breakdown of the complex molecule, and they produce some water molecule. Plus, they release CO2 in our atmosphere at a night time. Yes, guys, plant also respire. Plants, microbes, any kind of animal, all respire. All living thing can respire. This is very essential for life, guys. Very essential. Very essential. This is the daily activity. We can't survive without respiration. Let's start. Let's start with respiration. Here is some examples. Here is some examples that how a animal can respire, how a plant can respire. At a daytime they produce photosynthesis and produce food, and at night time they produce like CO2 and water molecules over there. So this is all about it. This is all about it. So the plant food name is C6H12O6. The plant food name is, it may be starch, it may be glucose, it may be sucrose. Like this is the form of carbohydrate. This is the synthesis form of the food. Glucose is synthesized form. The synthesis by glucose. Sucrose, this is the transporting phase. This is the transporting form of food. This is the synthesis form of the food. And star, this is the storage form of food. Storage form of food. If plant want to supply their food to all our body, they convert into sucrose. If plant want to synthesize the food, they synthesize the food in the form of the glucose. If plant wants to storage the food, they can store as a starch. That's all about it. Animals are heterotrophic. They does not synthesize anything. They obtain food from plants directly and indirectly. Saprophytes, what they do? Saprophytes like fungi are dependent on the dead and decay plant. They feed. What they feed? They already dead tissue eat, and they already decay matter will eat. Okay. Now, what is the important of recognize is that ultimately all the food that is required for the life process come from photosynthesis, guys. Yes, food will break down and we expire. Respire. This chapter deals with cellular respiration, or the mechanism of breakdown of food material within the cycle of release energy and the trapping of this energy for synthesis of ATP. Yeah. Here, how ATP will form. How ATP will form, we will study here. This is the definition of respiration. Look at here. What is the definition of respiration? The breaking of C-C bond, means carbon-carbon bond of complex compound through oxidation within the cell, leading to release of considerable amount of energy is called respiration. This is the important, remember this thing. The compounds that are oxidized during this process are known as a respiratory substrate. These substrate convert into their products. If food is substrate, then they convert into the water molecule and CO2, guys. Clear, guys? Let's start with some brief introduction, like how plant will breathe. Do plant breathe? Yes, of course. Here, look at here. Yes, but at a specific time, at a night time they respire, but in a daytime they don't respire. So yes, at a night time they respire. Okay. Plant require oxygen for respiration to occur, and they give out CO2 at a night time, guys. Plant have system in place that ensure the availability of oxygen. Yes, of course, they take oxygen and break down of the food carbohydrate products. Oh, I'm sorry, guys. Plants, like animals, have no specialized organ for gaseous exchanges. Yes, guys, we have lungs, but they don't have their high profile system. So they can respire with the help of stomata. They respire with the help of lenticles. The stomata found in where? At a leaf surface. At a leaf surface they are found. And where lenticels are found? They found stem parts. Small pore-like structure. What they do? They do some gaseous exchanges. Here also they do same thing. Look at here. This is a universal reaction for the respiration. Look at here. This is important, guys. Always remember this definition, always remember this reaction also. Clear, guys? Yes, clear. Now let's discuss about structure of stomata. Stomata structure, because this is in your course. This is the guard cell and this is the subsidiary cells, helper cells. They may be 4 to 6 in a number. It may be differ, okay? It may be differ. Where they located? At the surface of leaf. These are the guard cells, and these are the subsidiary cells. Now what guard cell contain? They also contain chloroplast. Chloroplast helpful in photosynthesis mechanism. Because of chloroplast, they can also helpful in photosynthesis. This is the pore. This is the, guys, look at here, this is the pore. With the help of this pore, they exchange and they release some gases molecule as well as water molecule. Yes, because of transpiration they release water, and because of intake of CO2 and exhale of oxygen they exchanges their gaseous molecule. Yes, and where lenticels are present, these are the stem, newborn stem. When stem that time is green, they contain small porous like structure at their bark, at the form of their bark, and with the help of these lenticels they exchange their gases. This is the reaction, guys. Remember this reaction every time. Listen to me, guys. This mechanism is not a wasteful mechanism, all right, that our food and oxygen molecule waste and we release CO2 and some water molecules. We found energy with them. Overall, we eat food and we have to break down this food. We eat food, and yes, the future is that food is breakdown. Tell me one thing, why we eat food? Why we eat food? Why we eat at a morning time, at a night time we eat food? Because we need energy, and energy form is ATP, guys. That's why we eat food, so we can get energy from this food. So we have to pay some oxygen, so we can intake oxygen, and here food will break down and give us to energy. So that's why we respire, guys. So respiration is most important phenomena. Here everything is written here. Because of ATP synthesis we respire, because we want energy. First formation of ATP by glycolysis. You know what, some special thing about glycolysis? They perform aerobically as well as they perform anaerobically. Means if oxygen is present, then glycolysis also perform their function. This process is occur, and if oxygen is not present, then also they can make their thing. If oxygen is present, still glycolysis occurring, and if oxygen is not present, then also glycolysis occurring, but they give less ATP, guys. Yes, if they take oxygen they give less ATP, and if they don't take oxygen they also give less ATP. Okay, let's start. Where this glycolysis process occur? Where? Cytosol, guys. Cytosol. Cell contain their cytoplasm. Okay, the glycolysis occur in cytoplasm of the cell. Who discovered this thing? Who give this chart? Yes, there are three scientists. G. Ambedon first, O. Meyerhoff second, J. Parnas third scientist. They discovered what? They write down the scheme of glycolysis. Okay, now what's special? We can call it as the EMP pathway. In anaerobic organism, it is the only process in respiration. Those animals who already anaerobic in nature, they don't take oxygen. So this is the life of anaerobic organism. Those organisms who respire without oxygen, but glycolysis, this process already occur even in absence of the oxygen, even in presence of the oxygen. In this process, glucose, look at here, glucose. Now synthesis form of the food by the plant is glucose. If they want to transport their food, plant If they want to transport their food, what is the form of the food? Sucrose. If they want to store their food, what is the form? Starch. So in this process, glucose undergo partial oxidation to form two molecules of pyruvic acid. A 6-carbon compound, glucose, break into 3-carbon compound, pyruvate acid. We can say pyruvic acid. 3-carbon compound, 1, 2, 3-carbon compound. But we have 2 molecules of 3 compound. So this thing is saturated. Here is 6-carbon balance. Here is also 6-carbon balance. But this breakdown take at least 10-step process, guys. This is not easy, guys. If we break anything in our body, they require lots of process. This is not a 10-step process, guys. This is multiple step process. But in our book, they complete the 10-step process. Anything, if we want to break inside the body, they take multiple process. This is not a one-way pathway. Guys. It takes lots of steps for breakdown. This form converts into this form. We thought, what is this? One bond will break and we get this molecule. No, this is not easy. Let's start, guys. Look at here. Sucrose is converted into glucose and fructose by the enzyme invertase. And these two monosaccharides readily enter to glycolytic pathway. Means sucrose converted into glucose and fructose with the presence of this enzyme. So they can perform this pathway. So glucose can perform this pathway. Let's start. Here is theoretical part and here is the flow chart. Remember now 10-step pathway. Quickly remember khatam. Glucose, this is the 6-carbon molecule. What happened with this? Glucose change into, wait a minute, let me change the pen. Glucose change into glucose 6-phosphate. Now this is the glucose. One phosphate group is added. So they can convert into glucose 6-phosphate. Is this easy? No. They give energy to conversion of this to this. Now this appearance is bad. They won't change their face. So glucose change into fructose 6-phosphate. Look at here, guys. Sucrose can convert into glucose as well as fructose. So this is not a difficult task with the presence of this enzyme. Now they change their face with the help of which enzyme? Invertase. And this performance is done by hexokinase. Okay, kinase means addition of phosphate group. Next, fructose 6-phosphate converts into fructose 1,6-biphosphate. Means again one phosphate group is added. What thing we pay? Energy. Again we lost energy. Means fructose again 6-carbon chain. This phosphate already is here, and again one. Means this is the position, guys. One here, one phosphate will attach. This is the position of phosphate, guys. At a first position, at a sixth position, phosphate group attach. At a sixth position, phosphate group attach. Now what happened next? This 6-carbon compound separate. This is separate. 3-carbon and 3-carbon. Alright, they were going to separate. 6-carbon chain will separate here, not end part. Here they separate. 6-carbon molecule, they separate, divided into 3-3-carbon. 3-3-carbon. I'm drawing here 3-3-carbon. 3-carbon, and here also a 3-carbon. Where is their phosphate group? Yes, they have their phosphate group, guys. Okay, now what happened with this? They can be interconvertible. So we can take both 2 into 3-carbon. We take this molecule also and we can take this molecule also because they can convertible form. Triose phosphate. We can say it as a glyceraldehyde 3-phosphate. Very important, guys, very important. When we study our C4 cycle, C3 cycle, this form is very important, star mark. This process need everyone. Everyone need this process, this molecule. Triose phosphate, dihydroxyacetone phosphate. What is a phosphate? Phosphate means phosphate group, guys. Phosphate means phosphate group. So these both molecules are interconvertible. Okay. Okay, now what happened to next? What happened to next? Now we take both of them, 3 molecule, both of them. Now I'm writing here this time. Look at it, 2 into 2 into 2 into 2 into 2 into 2 into 2 into, because we take both the molecule. Now this form, triose phosphate form, change into triose bisphosphate, 2-phosphate. Now 1,3-bisphosphoglyceric acid, and this form converts into triose phosphate, 3-phosphoglyceric, not glyceraldehyde, glyceric acid. This form very important according to our cycle. But this conversation convert into this conversation. We don't pay anything. We get energy. Yay. Get energy. Yes. This form converts into this form, 2-phosphoglycerate. And now 2-phosphoglyceraldehyde change into phosphoenol pyruvic acid. Yes, this form is also important in our C4C3 cycle, guys. Never forget this. Never ever. We will study this enzyme in PEP pathway. Okay, guys, let's move further. Phosphoenol pyruvic acid, they change into finally the end product is pyruvic acid. This. Finally, we get pyruvic acid, and this pyruvic acid goes into Krebs cycle. Guys. And after entering in Krebs cycle, the Krebs cycle also give us to ATP. Here also we get ATP. Here we get ATP. Here we get ATP. Yes, get energy. We also get a water molecule. Yes, na? What is the reaction, guys? This is the reaction, na? This is the reaction. And we have to find CO2. We release in atmosphere. And we also get H2O. Look at here, this is H2O. And we get energy. This is the energy form. So this equation is correct. So this equation is correct, guys. According to this reaction, we get this, and this. So this is perfect. This reaction is perfect, guys. Perfect. How much we lost? We lost 2 ATP of energy. We lost 2 ATP, and we get how much ATP? And we get 4 ATP. Why? Guys, 2 into 2 into 2 into 2. So into 2 here, 2 here. We get 4 ATP, and we calculate total. Then we get total 2 ATP. That's it. So we get ATP from glycolysis pathway, guys. Alright, clear? Very clear, guys, very clear. Look at here, guys. Here we also found some energy. This is the form of energy. So we also get energy in this form. So yes, glycolysis pathway provide what, guys? Energy. Yes. So this is the beneficial pathway. Look at here. Starting is ATP utilizing steps, here to here, here to here. ATP will utilize, and now next is the starting phase is preparatory phase, and this is the conversion phase. Okay, the second phase of conversion of fructose 6-phosphate to fructose 1,6-phosphate. Okay, the fructose 1,6-phosphate is split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. We know now. We elaborate this chart one by one. Whatever I explain here, everything is right here. Don't be panic. I everything explain here. How ATP will form, I also explain. Okay, in our next video, we will cover fermentation. So that's all for now. And if you still not subscribe my channel, then please subscribe. Thank you. Thank you again for listening me. Thank you so much.